Welcome to this educational video on biotechnology. Today, we will be exploring the basics of biotechnology, including the DNA cloning process and DNA analysis methods. First, let's start with an introduction to biotechnology. Biotechnology is the use of biological systems, organisms, or processes to create products or improve existing ones. It is a field that has rapidly grown in importance and has applications in medicine, agriculture, industry, and environmental conservation. One of the most important applications of biotechnology is the DNA cloning process. DNA cloning is the process of making multiple copies of a specific DNA sequence, which can be used for research or production purposes. The DNA cloning process involves several steps, including DNA extraction, PCR amplification, and transformation. DNA extraction is the first step in the cloning process, where DNA is isolated from cells using various techniques. The next step is PCR amplification, which is a process that allows for the amplification of a specific DNA sequence. The amplified DNA can then be inserted into a vector, such as a plasmid, which is a circular piece of DNA that can replicate in bacteria. The next step in the DNA cloning process is transformation, where the recombinant plasmid is inserted into bacteria. This process allows for the creation of many copies of the recombinant DNA, which can be used for research or production purposes. Now, let's move on to DNA analysis methods. DNA analysis methods are important tools in biotechnology, as they allow for the identification and analysis of DNA sequences. Some of the most common DNA analysis methods include gel electrophoresis, PCR, and DNA sequencing. Gel electrophoresis is a method used to separate DNA fragments based on their size. DNA fragments are loaded onto a gel and an electric current is applied, causing the fragments to move through the gel. Smaller fragments move faster than larger ones, allowing for the separation of DNA fragments based on their size. PCR, or polymerase chain reaction, is a method used to amplify a specific DNA sequence. This process involves the use of a DNA polymerase enzyme to amplify a specific DNA sequence through a series of heating and cooling cycles. DNA sequencing is a method used to determine the sequence of nucleotides in a DNA molecule. This method involves the use of DNA sequencing machines, which use various techniques to identify the sequence of nucleotides in a DNA molecule. In conclusion, biotechnology is a field that has rapidly grown in importance and has applications in various fields. The DNA cloning process and DNA analysis methods are important tools in biotechnology, as they allow for the manipulation and analysis of DNA sequences. Thank you for watching this educational video on biotechnology.